Chapter 18 is going to look at the different ways that gene expression is regulated. So remember, gene expression was this idea of DNA to RNA to proteins, which is what we did in chapter 17. So now we're going to look at ways where these steps are either turned on or turned off. And not all of our genes are expressed in every cell all the time. So we have to have ways of expressing the proteins we need and not expressing the ones that we don't need. That's what the regulation part is. So we're going to start with prokaryotes. Bacteria often have a single chromosome. It's a little bit simpler. And most of the genes for a specific function are going to be grouped together. And these are often grouped into segments that can be kind of turned on or turned off, called an operon. So here we can see a general structure of an operon. So structural genes X, Y, and Z are going to code for proteins X, Y, and Z that are going to have some function inside of the cell. They're typically going to either function together to make an item or all function in a similar function to the point where they would all be turned on or all be turned off at the same time. We know that the promoter region is going to be at the beginning of the gene transcript. So here's my promoter control region. So the promoter is where the transcription factors bind to start initiation of transcription. And there's another section in here called the operator. The operator is essentially the on-off switch. When the operator is on, that means that it's going to stimulate transcription of this gene, which is eventually going to express the genes X, Y, and Z. When the operator is off, it's going to turn this gene off, so this gene would not be expressed. The operator is operated by a protein called a repressor. So if you think about being repressed is kind of a negative word. It's something that's going to turn something off. So if a repressor is active, that means it's repressing the operon, and that's going to turn the operon off. If a repressor is inactive, that means it's not repressing, and that's going to allow the operon to be on. There's two different types or classes of operons that we see in bacteria. The first is called the repressible operon. So an example of this is the tryptophan, which makes tryptophan for bacteria. So a repressible operator is by default on. So these are going to be in the on position, and then they can be repressed or they can be turned off when they need to be. These are typically anabolic, which means they're going to be building something. In this case, we're going to be building tryptophan. So you can see the entire operon down here. We've got the promoter sequence. We've got the operator, which is right here. There's several genes, A, B, C, D, and E. All of these need to be made for tryptophan in order to be made. And right now, this tryptophan is absent. So by default, tryptophan is being made. So typically the cell needs tryptophan to be able to make more proteins. So at a default, when there's not very much tryptophan present, the operon is just on. The operator is not bound to the repressor. So you can see the repressor here is inactive because there's not a lot of tryptophan present. So this is the default situation. So what's going to happen is transcription is going to occur. We're going to make our proteins. This is eventually going to result in making tryptophan. When tryptophan is present, so if tryptophan is present, it is a co-repressor. So what it does is it binds here to the repressor, and that is going to activate the repressor. So tryptophan plus repressor, and you can see the shape change here from kind of bent to straight in a row. And you can see how this shape is going to fit right here in the operator. So when the repressor binds to the operator, it's going to turn it off. This becomes repressed. Hence, it is a repressible operon when tryptophan levels are very high. And then eventually, when tryptophan levels are low, this tryptophan will unbind, the repressor will go back to its inactive state, and then the operon can turn back on again. The other type of operon is called an inducible operon. When we think about inducing something, we think about kind of starting something or turning it on. So inducing labor is when we kind of get somebody started with labor if they weren't doing it themselves. So inducible is usually off. So these are typically off until we need them. And they're usually catabolic, which means we're going to be doing some kind of breaking enzyme. So the textbook inducible operon for bacteria is the lac operon. Lac stands for lactase. So if there is no lactose, lactose is a sugar that gets broken down by lactase. If there's no lactose, we don't need lactase to break it down. So that's why this gene is usually off. However, if you introduce lactose to a bacteria, it now needs an enzyme to be able to break that down. So it's going to be enzymes here. You can see LAC-Z, LAC-Y, LAC-A. These are the enzymes for using lactose. So these are going to be the enzymes that are going to allow that bacteria to process that sugar. So when the lactose is present, you can see here's the lactose. It's going to bind to the repressor, and it's going to turn it off. When it turns the repressor off, that means it's going to turn the operon on. So an inactive repressor allows the operon to function, and now I can make the enzymes that I need for processing the lactose. Once all of the lactose is processed, once it's absent again, then it gets removed from the repressor. The repressor is going to go back to binding the operator, and this is going to turn the entire operon back off because we don't have any more lactose that we need to process.